A very good morning to you all, and a good evening to folks joining from India. And thank you, Dr. Dewey, for that introduction. I couldn't say it better myself. Uh, I would also like to thank the Office of Interdisciplinary Graduate Programs for this recognition and the opportunity to share my work with the peers and friends alike. I hope you can all see my screen, and I'm going to be moving my mouse pointer for better illustrative purposes. So, okay. So today I'll be talking about what evolutionary genomics research is and how it is applied in a conservation context. And I'll be talking about how I've implemented that research concepts for the conservation of the Montezuma quail, this little cute bird that you're seeing on your screens right now. But before that, a bit of truth. We have enough evidence on global biodiversity loss that the present era is being labeled as the onset of the sixth mass extinction. And since the last ice age, the humans have colonized the entire globe and have massively changed the landscape. On the map you can see here shows the percentage of natural habitat loss since the Industrial Revolution. And most populated areas have lost most of their landscape. In the past 5,000 years, we have, during the global colonizations, we have extinct most of the megafauna, like the giant sloth you see here. But since the Industrial Revolution, more and more smaller and smaller species are also getting extinct or are currently threatened. A recent study showed that there has been a rapidly loss of populations in most of the species across all major continents, with many species losing up to 80% or more of their populations. So this actually paints a very grim picture, and there is a dire need for conservation because most of the biodiversity right now are facing threats due to extreme weather events, due to climate change, and loss of their habitat. So what does conservation research actually tells us? So one thing that it tells us that smaller populations are more prone to extinction. This might sound obvious, but nothing is obvious until proven to be obvious. And extinctions occur when species lose their evolutionary potential to adapt to these climatic or environmental stressors. And loss of genetic diversity compromises the ability of a species or population to adapt. So because populations that are less genetically diverse are more prone to diseases and infections. And we know this because of evolutionary genetics. So the evidence comes from evolutionary genetics, which is the synthesis of Darwinism and Mendelism, if that's even a word. But the idea that species evolve in response to their environment. One example, highly studied example, is the evolution of beaks in birds that have adapted to their dietary preferences. And we also know that these traits genes are responsible for these traits, and these traits are inherited during the process of reproduction. So what exactly is evolution? In the simplest of terms, evolution just means the changes that are inherited during reproduction. To understand this, let me show you an illustration that I found in a science textbook that my mom teaches to high school students back in India. So imagine that there is a population of red bugs. In first case, a green bug enters the population that is more adapt to hide in its environment, and crows are not able to find this and they eat the red bugs, and over time more and more red bugs are eaten, and green bugs who are more adapt are able to repopulate the environment. In this case, the evolution of red bugs to green bugs is called selection. In the other process, let's assume that a blue beetle or blue bug enters the population, but this time blue and red are equally as bad as to hide to their environments. But one day an elephant comes and tramples all the red bugs. This is a very extreme example, but highly plausible, and leaving behind the blue bugs to repopulate the environment. This is an example of demographic changes, sometimes called in the literature as drift. And in the third case, there is no new mutations entering, but the environment degrades. And as an adaptive measure, the get smaller, but once the habitat is restored, they're able to change back to their original forms. And this is an environmental adaptation. So in the first two cases are genetic responses, whereas the third one is purely environmental. So what exactly is genetics? Genetics is the study of genes and genomes. And we all know that all life on Earth is gene-based, and every species have genomic DNA within their beings. And Mendel showed in the 1800s that these phenotypes or these changes we see are based on the genes that are inherited by the mixing during reproduction. So genomics, now what is genomics? Genomics means that the study of the whole genome, and the genome is the complete set of DNA, which is found in the nucleus of almost every cells within the body. And they are contained within the chromosomes. And if you keep unwinding them, then you get this large strand of DNA that is made up of base pairs A, T, G, and C. So there are only four letters and their combinations that comprises the entire genome. So population genomics. So what is population genomics? It's the study of the genome from multiple individuals representing different populations. So a genome contains genes, but it also contains intergenic regions that were previously called junk DNA, but we are learning more and more about its evolutionary it causes its evolution. So what is population genomics? Like I said, when we compare the genomes of multiple individuals and look at the differences. So in the most basic example that includes birds and mammals, the genome is inherited as two diploid chromosomes. So each individual has two sets of this DNA, one coming from the mother, the other coming from the father. And if you compare these, you can look at these variants or mutations that are difference that causes the differences between the individuals and populations. We can look at where these mutations occur within the genome, and we call this as SNP, single nucleotide polymorphism, but think of this as just a new mutation or a variant or a marker. And by looking at where these mutations occur within the genome and at what frequencies they exist. So for example, in this case, G and C are equally likely within the population with 0.5 of frequency each. So by understanding these allele frequencies and where these mutations occur, we can get the evolutionary history of the populations. So with that introduction, I'll now switch gears to talk about Montezuma quail. So what do we know about Montezuma quail? Well, for starters, we know that we don't know much about it because it's one of the least studied bird species of North America. And just like chickens and pheasants, these quail are also ground-dwelling birds, which means that they don't migrate or disperse long distances. And they're mostly found at higher elevations between 4,000 to 5,000 feet in the oak grassland and desert grassland landscape, mostly in Mexico, but they have populations coming up to southwestern United States with a more continuous population in Arizona, little bit fragmented population in New Mexico, and highly fragmented and isolated populations in Texas. One of the problems why we don't know much about it is because it's extremely difficult to trap and monitor, because these quails are very well camouflaged in their environment and they like to hide in tall grasses, and that's why we don't know much about it, because we cannot find it, and if we do find it, we're not able to monitor it. And just because, as you can see from the map, the populations in Texas are very isolated, and they have been declared as vulnerable by the state of Texas. So how do we actually conserve a population or a species? There is a conservation genetic framework, and the first question we have to ask is, are there multiple populations? And if there are not multiple populations, then we have to address the non-genetic or the environmental issues. But if those populations are distinct, are they isolated? And if they're isolated, are the isolated population undergoing inbreeding? And if not, then we have to increase the population's sizes. But if they are isolated, distinct, and inbred, the question comes in, will crossing of smaller populations with larger populations is going to help the smaller populations? If not, then we have to preserve the local habitat where the isolated population exists. But if crossing would be beneficial, then what would be the optimum strategy to do so? So within this framework, I've based my dissertation research. So I started at the very basic, where was the whole genome sequencing. So I extracted the DNA from genomic DNA from one male and one female Montezuma quail, and I sequenced the genome, assembled the genome. I annotated, which means found out where the genes exist within the genome, and found these variant markers or the SNP markers. So in short, now we have the whole genome of Montezuma quail in the literature, and we also have an idea of where all these mutations occur within the genome. And Montezuma quail genome size is 1.3 gigabases, which means it has 1.23 billion ATGC letters within the genome. So just to get a preliminary idea of the population genetics of these US populations, what I did was, out of these all the variants of mutations, I handpicked mutations from that existed in the genes. And I chose these genes because they were known to have a crucial role in biological processes like reproduction, growth, immune response. And I created a diagnostic panel using these genes, using these mutations that existed in the genic regions. And I also chose some SNPs that existed outside the genic regions and created this diagnostic panel that can be used by wildlife conservation managers who need to monitor these birds. And I used this panel to get the genotypic or genetic information for 188 Montezuma quail individuals. To answer the first question, are there multiple populations? And the data suggests that they all form independent clusters. So to provide you a little more context, what exactly this means, this is a similar analysis done with human populations. And this suggests that the European population is very closely related to the Middle Eastern population, and they share genetic similarities. Similarly, the South and Central Asian populations have shared ancestry with the European populations, but at the same time, some of these populations also share ancestry with East Asian populations. And Native American populations are closely related to the East Asian, whereas the Oceanic Islander populations form their isolated independent clusters. And the most distinct of them all are the African populations, which are the most ancestral to the human species. So our data suggests, given that they are ground-dwelling, non-migratory birds with isolated population in Texas and their genetic independence, that these populations are isolated. So to dig more about this fragmentation and isolation and the genetic signatures of it, I studied the three populations in the US, Arizona, New Mexico, and Texas, and sequenced 90 whole genomes of 90 different individuals. So I analyzed more than 250 billion bases, these A, T, and G, and C, and looked at more than 6.6 million mutations that exist within the genome. And I also did that. So when I say whole genome, I mean I'm looking at mutations both in the genes and the intergenic regions, but I also exclusively looked at the genes and the mutations that exist in these genes. And Montezuma quail has more than 17,500 genes, and variation in these genes is a proxy of adaptive diversity, because fundamentally it is predicted that genes are the ones that are going to change the phenotypic behavior that is responsible for the evolution or adaptation. So what does our data suggest us? Our data suggests us that the Texas population have significantly lower whole genome diversity as compared to the larger and intermediate sized populations of of Arizona and New Mexico. And we saw a similar reduction in diversity in the genic or the adaptive regions. And to put more context about this, the Texas Montezuma quail are the least diverse of them, and their genomic diversity is comparable to other avian species of conservation concern, like the white-tailed eagle and the bald eagle. So yes, so now the question is that, yeah, there are multiple populations of Montezuma quail, and Texas population is isolated. But the question is, are they inbred? Well, inbreeding is actually a problem because inbreeding reduces the fitness. And as an example, think about a father who is heterozygote for these two genes. This gene having one copy of the good gene and one copy of the bad gene. And since he has good gene, he is unaffected. Same is the case for the mother. And they mate and reproduce and create these three progeny who are all unaffected because they have at least one copy of the good gene. But there's a 25% probability that these both bad genes are gonna become homogeneous within a certain individual, and this could lead to expression of the bad gene, which was being repressed by the good gene. We call these kind of mutations, bad mutations that homogenize in the individual, as homozygous recessive. And if the populations are inbreeding, these kind of homozygous recessive mutations rise in the frequency, and it could be bad for the fitness. So to look at whether these populations, especially Texas, are inbred, I looked at the individual genomes and looked at whether they were homozygous, as in this case, it's a CC homozygous versus heterozygote, TA heterozygote. So if the population or individuals are more inbred, they'll have more of these homozygous sites and less of these heterozygous sites. And our data indeed suggests that on average, Texas Montezuma quail are significantly more inbred than the larger and the intermediate size populations. We also looked at the ratio of these homozygous to heterozygous sites. And again, we saw that Texas quail have more homozygous sites than heterozygous sites, where this dotted line represents equal proportions of these sites. So the question comes, so inbreeding itself is not bad, but many of the species actually are inbred to a certain degree. But these, as I previously mentioned, inbreeding becomes worse when these homozygous recessive mutations rise in the frequency. So this is a schematic of how actually the gene looks that has coding and non-coding regions. So I looked at the mutations that existed in all of these genes, 17,000 genes in the Montezuma quail genome, and classified these mutations as high impact, moderate impact, low impact, or non-coding, with high impact mutations predicted to be the worst for the gene, and low and non-coding impact might be less detrimental or less harmful for the gene. And as predicted, we actually found that Texas quail have more higher frequencies of these high impact and moderate impact and even the low impact mutations, which means that these bad mutations that arose in the population got homogenized in the Texas quail because they were inbred and resulted in the higher frequencies. And this is actually pretty bad for the Texas quail because this means that Texas Montezuma quail are most at risk of inbreeding depression. So inbreeding depression just means that the fitness of the individual or population at large is worse because of inbreeding. And there's evidence to show that a lot of species that got extinct, including woolly mammoth and our cousins, the Neanderthals, got extinct because of inbreeding depression. And we have documented this case for the loss of a lot of populations for the isolated populations of wolves and Florida panthers. And anyone who has seen Game of Thrones would attest to the fact that inbreeding can also be a problem in human populations. So the question comes that, yes, Texas quail are independent, are isolated, and also inbred, but would crossing with the larger Arizona population would be beneficial And if yes, what would be the best strategy? So we call this process of moving individuals from one population to other as translocations. And if this will work in Montezuma Quail is literally $1 million question, because translocations are costly and stressful. So imagine moving to a different locations and to a new environment, and besides adapting to that new environment, finding someone to mate with so that the population actually gets better. And it's even worse for wildlife because it requires a lot of effort, ground effort from people who are outside academia and wildlife conservation managers. So answering this question is very important so that the resources can be allocated according to the priorities. So this question, again, like I said, is not easy to answer because this is something that is not happened and would be in the future. So the idea of these crossing or translocation is to maximize the adaptive potential of a small populations, and adaptive potential is dependent on adaptive diversity, which is the genetic diversity. Higher genetic diversity means higher adaptive potential, and the mutation load of these bad homozygous recessive mutations, we have to minimize that to maximize the adaptive potential. So in Montezuma quail, there is a large population in Arizona, and there's even possibly even larger population in Mexico. And then there is isolated small population in Texas. So what I'm trying to estimate is if periodic translocations from these larger populations would be more beneficial to improve the adaptive potential of these small isolated populations. So we don't know this yet, but I'm trying to figure this out using more than 100 whole genomes from the 3 populations in the US. And now we also have samples on whole genomes from Mexico populations. So how am I doing this? I'm doing this based on demographic-based simulations of genome in forward time. So think about this way. We have to figure out the history of these populations to predict how the future is going to look. So demographic-based means to understand back in time when these populations were a single population, and since their time of their departure, how much they have diverged. And understanding these questions is important because these parameters of how big the populations were and how long they have diverged for are important, because these parameters are going to be used to predict the future changes. And I'll be running simulations mainly with 2 translocation models. The first being no simulation, no migration, which means that we have to see how much these populations are going to diverge and how worse the Texas population is going to be. And then the other would be with different migration rates, where we are actively translocating individuals from one population to the other, which is in the literature is called as a genetic rescue measure. So the most optimum genetic rescue strategy would be that maximizes the adaptive potential based on measuring these 2 metrics and minimize inbreeding, and have to do that in the least number of generations. And I am doing this right now as we speak. Is I'm running simulation models and demographic models, and hopefully I'll be able to share this research with the public soon. And so just to summarize my talk, I hope that I've shown you that how evolutionary genomics can help us understand the adaptive potential of species, especially if they are of conservation concern, and how we can use genomics to create these genetic monitoring and diagnostic tools that can be readily used by land managers and wildlife conservation managers who actually go out in the field to monitor these species. And I hope that I've shown you that fragmentation and isolation reduces the genomic diversity and increases inbreeding. And a long-term conservation effort should maximize the adaptive potential of small populations and hopefully minimize the inbreeding depression. And with that, I would like to acknowledge Purdue and all the affiliated departments that I work with. I want to also thank all the partnering universities and state agencies that have provided us with these samples for the genomic analysis. And I want to thank our academic collaborators. I also want to thank our funding agencies, and extra thanks to Welder Wildlife Foundation that graciously sponsors my PhD fellowship. And a huge thanks to Dr. Andrew Divoody, my PhD advisor, who not only accepted me to be a part of his team, but also has painfully mentored me throughout these years. And a shout out to all the friends and family members that have made along the way of my life's journey, and extra love to my parents who are here and my family members who have supported me throughout these years. And I would like to thank everyone who joined today, and I really appreciate your time and effort. And to conclude my talk, I leave this video that simplistically shows how evolution works as I take questions from the audience. Thank you all.